So clear. So today our topic is bacterial photosynthesis. So our topic is bacterial photosynthesis. So bacteria are based on photosynthesis. The bacteria are classified into three major groups. So based on photosynthesis, the bacteria are classified into three major groups. So based on photosynthesis, the bacteria are classified into three major groups. They are green sulfur bacteria, next one is the purple bacteria, and third one heliobacteria. So based on photosynthesis, the bacteria are classified into three major groups. They are green sulfur bacteria, purple bacteria, purple bacteria and heliobacteria. So first of all, I am going to discuss about green sulfur bacteria. Green sulfur bacteria. So first group is green sulfur bacteria. The green sulfur bacteria, they belongs to Chlorobacteriaceae. They belongs to Chlorobacteriaceae. So examples of green sulfur bacteria. The common one is the chlorobium, and chloroseudomonas. So clear. The green sulfur bacteria, they belongs to Chlorobacteriaceae. The most common green sulfur bacteria are chlorobium, and chloroseudomonas. So in this bacteria, in green sulfur bacteria, chlorosomes, not chromosomes. In green sulfur bacteria, chlorosomes are the photosynthetic operators. So chlorosomes are photosynthetic. So photosynthetic operators. So clear. So the green sulfur bacteria, they belongs to Chlorobacteriaceae. The common green sulfur bacteria are chlorobium and chloroseudomonas. In this bacteria, chlorosomes. So chlorosomes are photosynthetic operators. The chlorosomes are vesicle-like structures. The chlorosomes are vesicle-like structures. So chlorosomes are vesicle-like structures vesicle-like structures closely associated with. So closely associated with the cytoplasmic membrane. So closely associated with the cytoplasmic. So cytoplasmic membrane. So cytoplasmic membrane. So chlorosomes are photosynthetic operators in green sulfur bacteria. These are vesicle-like structures. So vesicle-like structures closely associated with cytoplasmic membrane. Cytoplasmic membrane by they are closely associated with cytoplasmic membrane by crystalline basal plate. So crystalline associated with cytoplasmic membrane by crystalline basal plate. Crystalline basal plate. So clear. So green sulfur bacteria, they belongs to Chlorobacteriaceae. The common green sulfur bacteria are Chlorobium and Chloroseudomonas, etc. In green sulfur bacteria, chlorosomes are photosynthetic operators. So chlorosomes are vesicle-like structures. So vesicle-like structures, they are closely associated with cytoplasmic membrane by crystalline basal plate. So crystalline basal plate. So clear. The chlorosomes are surrounded by. The chlorosomes are surrounded by. The chlorosomes are surrounded by. The chlorosomes are surrounded by single layer. The chlorosomes are surrounded by single layer of galactolipids. Galactolipids. They are surrounded by single layer. They are surrounded by single layer of galactoproteins. Single layer of galactoproteins. So next one, the chlorosomes. The chlorosomes consist of rod-like structure. The chlorosomes consist of rod-like structure. This rod-like structure is known as bacterial. So bacterial chlorophyll C. Chlorophyll C. Chlorophyll C protein complex. Protein complex. Bacterial chlorophyll C protein complex. So next one, the cytoplasmic membrane. The cytoplasmic membrane consists of non-crystalline. So the cytoplasmic membrane, it consists of non-crystalline bacterial. So bacterial chlorophyll A protein complex. So clear. The chlorosomes consist of rod-like structure is known as bacterial chlorophyll C protein complex. Next one, the cytoplasmic membrane. The cytoplasmic membrane consists of non-crystalline bacterial chlorophyll A protein. So clear. So in green sulfur bacteria, the reaction center is P840. So P840 is the reaction center. So reaction center of green sulfur bacteria. In green sulfur bacteria, the reaction center is P84. In PS1, it is P700. In PS2, the reaction center is P680. But in green sulfur bacteria, the reaction center is PIT4. So clear. This is the information about green sulfur bacteria. The green sulfur bacteria belongs to Chlorobacteriaceae. The common green sulfur bacteria are Chlorobium and Chloroseudomonas. So chlorosomes are photosynthetic operators in green sulfur bacteria. These are vesicle-like structures. Closely associated with the cytoplasmic membrane by crystalline basal plate. So crystalline basal plate. The chlorosomes consist of rod-like structure is known as bacterial chlorophyll C protein complex. The cytoplasmic membrane consists of non-crystalline bacterial chlorophyll A protein complex. In green sulfur bacteria, the reaction center is P840. So clear. This is the information about photosynthetic bacteria, green sulfur bacteria. And next one is the purple bacteria. So next photosynthetic bacteria is purple bacteria. So next one is the purple bacteria. These purple bacteria are again classified into. These are again classified into two types. The purple bacteria are again classified into two types. They are purple sulfur bacteria. And next one, purple non-sulfur bacteria. The purple bacteria are again classified into. Two types. They are purple sulfur bacteria and purple non-sulfur bacteria. So clear. The first one is the purple non-sulfur bacteria. So purple sulfur bacteria. So first one is the purple bacteria are classified into two subgroups. They are purple sulfur bacteria and purple non-sulfur bacteria. So first of all, purple sulfur bacteria. The purple sulfur bacteria, they belongs to Chromatiaceae. They belongs to Chromatiaceae. The common purple sulfur bacteria are Chromatium. So Chromatium and Thiospirillum. These are most common type of purple sulfur bacteria. The purple sulfur bacteria belongs to Chromatiaceae. The most common purple sulfur bacteria are Chromatium and Thiospirillum. So next one, they use hydrogen sulfur. They use hydrogen sulfur as electron donor. So electron donor, they use hydrogen sulfur as electron donor. So this is the important one. The purple sulfur bacteria use hydrogen sulfide as electron donor. And the second group is purple non-sulfur bacteria. Purple non-sulfur bacteria. The second group is purple non-sulfur bacteria. The purple non-sulfur bacteria belongs to Rhodospiralaceae. Rhodospiralaceae. They belongs to Rhodospiralaceae. The purple sulfur bacteria belongs to Rhodospiralaceae. So the common purple non-sulfur bacteria, sir. The common purple non-sulfur bacteria are Rhodospirillum, and Rhodopseudomonas. Rhodopseudomonas. They are the most common type of purple non-sulfur bacteria. They are most common type of non-sulfur bacteria. They use alcohols. They use alcohols and carbonic acid. Carbonic acid as electron donor. The electron donors in purple non-sulfur bacteria are alcohols, carbonic acids. So alcohol and carbonic acids are electron donors in purple non-sulfur bacteria. So clear. The purple bacteria are again classified into two types. They are purple sulfur bacteria and purple non-sulfur bacteria. The purple sulfur bacteria belongs to Chromatiaceae. The most common bacteria are Chromatium, Thiospirillum, etc. They use hydrogen sulfur as electron donor. The purple non-sulfur bacteria belongs to Rhodospiralaceae. The most common bacteria are Rhodospirillum and Rhodopseudomonas. They use alcohol or carbonic acid as electronic donor. So clear. In both sulfur and non-sulfur bacteria, both sulfur and non-sulfur bacteria, chromatophores. So chromatophores So chromatophores are photosynthetic operators. So photosynthetic operators in purple bacteria is chromatophores. So chromatophores are photosynthetic operators in both sulfur bacteria and non-sulfur bacteria. So the chromatophores are vesicle-like structures. The chromatophores are vesicle-like structures. These are vesicle-like structures and originated from cytoplasmic membrane, originated from cytoplasmic membrane by invagination. They are originated from cytoplasmic membrane. The chromatophores are originated from cytoplasmic membrane by invagination. So chromatophores అనేది membrane, cytoplasmic membrane అనేది చొచ్చుక రావడం వల్ల ఏర్పడతాయి. So chromatophores are vesicle-like structures. They are originated from cytoplasmic membrane by invagination. The chromatophores also consist of a rod-like structure. The chromatophores also consist of a rod-like structure is known as bacterial chlorophyll. Bacterial chlorophyll. C. Chlorophyll. B. Protein complex. So clear. The chromatophores consist of a rod-like structure is known as bacterial chlorophyll B protein complex. Clear. So in purple bacteria, 870. So 870 is the reaction center. So 870 is the reaction center in both purple sulfur bacteria and non-sulfur bacteria. So clear. In green sulfur bacteria, the reaction center is 840. In purple sulfur bacteria, the reaction center, purple sulfur and both and non-sulfur bacteria, the reaction center is 870. So 870 is the reaction center. So this is the second group of bacteria. The first group is green sulfur bacteria and second one. So second one is the purple bacteria. So clear, the purple bacteria are classified into two types. They are purple sulfur bacteria and purple non-sulfur bacteria. They belongs to Chromatiaceae. So common ones are Chromatium and Thyrospirillum. They use hydrogen sulfur as electron donor. In purple non-sulfur bacteria, they belongs to Rhodospiralaceae. The common bacteria are Rhodospirillum and Rhodopseudomonas. They use alcohols and carbonic acids or electron donors. Both in sulfur and non-sulfur bacteria, the photosynthetic operators are chromatophores. The chromatophores are vesicle-like structures. So vesicle-like structures originated from cytoplasmic membrane by invagination. They consist of rod-like structure is known as bacterial chlorophyll B protein complex. The reaction center of purple bacteria is 870. 870 is the reaction center of 870 is the reaction center of purple bacteria. So reaction center of purple bacteria. So clear. And third one. So third group. The third group of bacteria. The third group of photosynthetic bacteria is heliobacteria. The third group is heliobacteria. The third group is called heliobacteria. Heliobacteria. So heliobacteria, they do not show chromatophores. Chromatophores and chlorosomes. So chlorosomes and chromatophores both are absent in heliobacteria. The pigments are, the photosynthetic pigments are present in cytoplasm. The photosynthetic pigments are present in cytoplasm. So clear, they do not show chromatophores. They do not show chromatophores and chlorosomes. The photosynthetic pigments are present in cytoplasm. The photosynthetic pigments are present in cytoplasm. So the heliobacteria consist of bacterial chlorophyll G. So this is the important one, bacterial chlorophyll G. Chlorophyll G. The heliobacteria consist of bacterial chlorophyll G. It is recently discovered. So the bacterial chlorophyll G is recently discovered. Clear? It is recently discovered. In heliobacteria, the reaction center is B798. B798 is the reaction center. B789 is the reaction center. Reaction center in heliobacteria. So clear. This is the information about types of photosynthetic bacteria. The photosynthetic bacteria are classified into green sulfur bacteria, purple bacteria, and heliobacteria. So the reaction center in green sulfur bacteria is B840. In purple bacteria, the reaction center is B870. So B870. In heliobacteria, the reaction center is B798. So clear. So what is the important? What is the key points? So one of the most important key points. One of the most important key points in bacterial photosynthesis. So key points. So key points in bacterial photosynthesis. So what are the key points? The first one of the most important one, the bacterial chlorophyll. The bacterial chlorophyll absorb. The bacterial chlorophyll absorb near infrared infrared region infrared region of spectrum of the spectrum. So near infrared region of spectrum. So it is known as invisible light. Invisible. Generally, the plant absorbs the visible light, but the bacteria are absorbed near infrared region of the spectrum. So there is no red drop. There is no red drop. So this is the another important one. The bacterial chlorophyll generally absorbed near infrared region of the spectrum. So no red drop occurred. No red drop occurred. And next one, in Rhodospirillum rubrum. Rhodospirillum. Rhodospirillum rubrum. So Rhodospirillum rubrum. In Rhodospirillum rubrum, the bacteria Rhodospirillum rubrum use the Kelvin cycle. It used the Kelvin cycle. The Rhodospirillum rubrum use the Kelvin cycle to synthesize carbohydrates. To synthesize carbohydrate. To synthesize carbohydrates. Carbohydrates. The Rhodospirillum rubrum use the Kelvin cycle for synthesis of carbohydrates. So clear. The bacteria like chlorobium. The bacteria like chlorobium. Chlorobium and chromatium. 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 The bacteria like chlorobium and chromatium use reductive carboxylic cycle. So reductive carboxylic cycle. Carboxylic. Reductive carboxylic cycle. Reductive carboxylic cycle to synthesize carbohydrates. So clear. So important one. One is the Rhodospirillum rubrum. It used the Kelvin cycle to synthesize carbohydrates. The Rhodospirillum rubrum used the Kelvin cycle to synthesize carbohydrates. The bacteria like chlorobium and chromatium use the reductive carboxylic cycle to synthesize carbohydrates. So clear. So next one, in bacteria, PS2 is absent. So PS2 is absent. So PS2 photosystem 2 is absent in photosynthetic bacteria. So there is no acyclic photophosphorylation. There is no acyclic photophosphorylation except cyanobacteria. Except cyanobacteria. So clear. In bacteria, photosystem 2 is absent. So there is no acyclic photophosphorylation except cyanobacteria. So cyclic photophosphorylation. So cyclic photophosphorylation is an important method. Important method of photophosphorylation or photosynthesis. So cyclic photophosphorylation is an important method in photosynthesis because PS2 is absent. So there is no acyclic photophosphorylation except cyanobacteria. In photophosphorylation, generally 2 ATP molecules are produced in each turn of cyclic photophosphorylation. Generally, in all plants, only 2 ATPs are produced in each turn of cyclic photophosphorylation. 2 ATPs are produced in all plants. But in bacteria, only 1 ATP, only one ATP produced in each turn of cyclic photophosphorylation. So each turn of cyclic photophosphorylation, only one ATP produced. This is the one of the most important one. So clear. So what are the most important key points in bacterial photosynthesis? The bacterial chlorophyll absorbs near infrared region of the spectrum. So invisible light. So no red dot. There is no red dot. Rhodospirale and rubrum use the Calvin cycle to synthesize carbohydrates. The bacteria's chlorobium and chromatium use reductive carboxylic cycle to synthesize carbohydrates. In bacteria, photosystem 2 is absent, so there is no acyclic photophosphorylation except cyanobacteria. So cyanobacteria is the only one bacteria perform the acyclic photophosphorylation because of presence of photosystem 2. So cyclic photophosphorylation, there is no PS2. So it consists of only PS1. So cyclic photophosphorylation is an important photophosphorylation method. In bacterial cyclic photophosphorylation, only one ATP molecule is produced each turn of cyclic photophosphorylation. Generally in plants, 2 ATP molecules. So 2 ATP molecules produced in each turn of cyclic photophosphorylation. In bacteria, only one ATP molecule is produced. So clear. So next one, another key point. So another key point is the reaction center. The reaction center of bacteria. The reaction center of bacteria is B890. B890 is the reaction center of bacteria. In bacteria, the reaction center is B890. In PS1, generally in PS1 is the P700 in plants. In eukaryotic plants, the reaction center P700 in PS1, PS2 is the 6CIT. But in bacteria, the reaction center is B890. B890. So clear. Next one, in bacterial photosynthesis, oxygen. In bacterial photosynthesis, oxygen does not produce, does not evolve because hydrogen donor is not water. The hydrogen donor is not water. In bacteria, the hydrogen donor is not water. So oxygen does not evolve. So what is the hydrogen donor? In bacteria, hydrogen sulfide. So hydrogen sulfide is the hydrogen donor. So hydrogen sulfide is the hydrogen donor. Hydrogen donor. So sulfur is evolved. So sulfur is evolved in bacterial photosynthesis because in bacteria the hydrogen donor is not water. The hydrogen donor is not water. The hydrogen donor is hydrogen sulfide. So hydrogen sulfide is the hydrogen donor in bacteria. So sulfur is evolved. In normal plants, the hydrogen donor is water. So oxygen is evolved. But in bacteria, hydrogen donor is not water. It is hydrogen sulfide. So water is not, sorry, oxygen is not evolved. Sulfur is generated. So clear. And finally, the photosynthesis in bacteria, the photosynthesis even occur in electric light. In electric light, generally, in electric light, the photosynthesis is absent, but the bacteria perform the photosynthesis even in electric light. Even in electric light, the photosynthesis is occurred in bacteria. So here, this is the information about bacterial photosynthesis. So bacterial photosynthesis. So clear. So once again, I repeat again. The photosynthetic bacteria are classified into three major groups. They are green sulfur bacteria, sulfur bacteria, and heliobacteria. The green sulfur bacteria belongs to Chlorobacteriaceae. The common bacteria are Chlorobium and Chloroseudomonas. In green sulfur bacteria, the photosynthetic operators are chlorosomes. Photosynthetic operators are chlorosomes. They are closely associated with cytoplasmic membrane by a crystalline basal plate. It consists of a rod-like structure. Is known as bacterial chlorophyll C protein complex. Next one, the cytoplasmic membrane consists of non-crystalline bacterial chlorophyll A protein complex. The chlorosomes are vesicle-like structures. They are surrounded by single layer of galactolipids. In green sulfur bacteria, the reaction center is P84 or B84. The purple bacteria are again two types. One is the purple sulfur bacteria and purple non-sulfur bacteria. Purple sulfur bacteria is the hydrogen donor as electron donor. So in purple non-sulfur bacteria, alcohols and carbonic acids are the electron donors. Both in sulfur and non-sulfur bacteria, the photosynthetic operators are chromatophores. So chromatophores are vesicle-like structures originated from cytoplasmic membrane by invagination. They consist of rod-like structure is known as bacterial chlorophyll B protein complex. In purple bacteria, the reaction center is B870. So B870 is the reaction center in purple bacteria. In heliobacteria, so heliobacteria do not show chromatophores and chlorosomes. The photosynthetic pigments are present in cytoplasm. So the main photosynthetic pigment is bacterial chlorophyll G. It is recently discovered. In heliobacteria, the reaction center is B798. The main important pathways of bacterial respiration, bacterial photosynthesis, they absorb near infrared region of the spectrum. So no red drop. Protospilum rubrum use the Kelvin cycle to synthesize carbohydrates. The chlorobium and chromatium use the reductive carboxylic cycle to synthesize carbohydrates. In bacteria, PS2 is absent, so there is no acyclic photophosphorylation except cyanobacteria. So cyanobacteria is the only one bacteria to perform the cyclic photophosphorylation because of presence of PS2. So in bacteria, cyclic photophosphorylation is an important phosphorylation method. In bacterial cyclic photophosphorylation, only one ATP molecule produced each turn of cyclic photophosphorylation. But in higher plants, two ATP molecules are produced each turn of cyclic photophosphorylation. So clear. And next one, the reaction center of bacteria. The reaction center of bacteria is B890. B890 is the one of the most important one. B890 is the reaction center of the bacteria, bacterial chlorophyll. In bacteria, in bacterial photosynthesis, oxygen does not evolve because the hydrogen donor is not water. The hydrogen donor is hydrogen sulfide. So, sulfur is evolved. In bacteria, the bacteria even the in bacteria the photosynthesis even occur in electric light. Normally, in higher plants, the photosynthesis not occurred in electric light, but in bacteria, photosynthesis even occur in electric light. So clear. This is the information about the bacterial photosynthesis. So this is the information about bacterial photosynthesis. So clear. The final topic of photosynthesis is factors affecting. So factors affecting photosynthesis. The final topic of photosynthesis is factors affecting. So factors affecting photosynthesis. So the scientist Sachs proposed concept of cardinal point. So concept of cardinal point. According to this theory, according to this cardinal point, factor affecting any physiological reaction. The factor affecting any physiological reaction has three main values. According to cardinal point, the factor affecting any physiological reaction has three main values. Three main values. The three main values are minimum. Any factor affecting any physiological reaction has three main values. They are minimum and optimum and a maximum. So according to cardinal point, the factor affecting any physiological reaction has three main values. They are maximum, optimum and minimum, or minimum, optimum and maximum. So clear. So first of all, minimum. The physiological reaction in minimum, the physiological reaction does not occur. The physiological reaction does not occur if the value is below minimum. The physiological reaction maximum, the value beyond the maximum. the value behind the maximum, the value behind the maximum, the activity is stopped. The activity is stopped. So clear. So this is the information about cardinal point. The cardinal point concept is proposed by Sachs. So Sachs proposed a concept of cardinal point. So according to this cardinal point, factor affecting any physiological reaction has three main values. They are minimum, optimum and maximum. Reaction does not occur if the value below the minimum. So below the minimum reaction not occurred. In optimum, the reaction can occur maximum speed. In maximum, the value beyond the maximum, the activity is stopped. So clear. So the factors affecting photosynthesis. The factors of photosynthesis, the photosynthetic factors, the photosynthetic factors, the photosynthetic factors, the photosynthetic factors are classified into the factors affecting photosynthesis. The factors affecting photosynthesis is classified into two major groups. They are external and internal. The photosynthetic factors are classified into two major groups. They are external and internal. The external factors, it includes, the external factors includes, the external factors includes light. The external factors includes light. The external factors includes light, concentration of carbon dioxide, and concentration of oxygen, and temperature, water and minerals. The internal factors. So the photosynthetic factors are classified into two major classes. They are external photosynthetic factors and internal photosynthetic factors. The external photosynthetic factors includes light, concentration of carbon dioxide, and concentration of oxygen, temperature, water and minerals. The internal factors. The internal factors include chlorophyll content, and protoplasmic factors, protoplasmic factors, and accumulation of products, accumulation of products, so accumulation of products, anatomy of leaf, etc. They include the internal factors. The photosynthetic factors are classified into two major groups. They are external and internal. The external photosynthetic factors include light, concentration of carbon dioxide and concentration of oxygen, temperature, water, minerals. So minerals, etc. The internal factors are chlorophyll content, protoplasmic factors, accumulation of products, and anatomy of leaf, etc. So clear. The photosynthetic factor. So first of all, I am going to discuss about external factors. So external factors of photosynthesis. The external factors of photosynthesis. In external factor of photosynthesis, the first one is the light intensity. So first one is the light intensity. Kanti Tivrata. So first one is the first external factor of the photosynthesis, light intensity. The light intensity, the light intensity at which rate of photosynthesis is equal to the rate of respiration is called compensation point. So clear. The light intensity at which the rate of photosynthesis, the rate of photosynthesis is equal to the rate of respiration is called compensation point. So clear. The light intensity at which rate of photosynthesis is equal to the rate of respiration is called compensation point. Compensation point. If light intensity, the light intensity, if the light intensity is increased, the light intensity is increased above compensation point, above compensation point, the rate of photosynthesis, is increased. So first one, if light intensity is increased above compensation point, the rate of photosynthesis is increased. So clear. So what is the compensation point? Light intensity, at which rate of photosynthesis is equal to rate of respiration is known as compensation point. If the light intensity is increased above compensation point, above the light intensity increased above compensation point, the rate of photosynthesis is also increased. But in presence of high intensity light, so in presence of high intensity లేదా light intensity, but in the presence of high light intensity, the chlorophyll molecules, but high intensity light, the chlorophyll molecules are degraded or destroyed. This process is known as photooxidation. So clear. If light intensity is increased above compensation point, if light intensity is increased above compensation point, the rate of photosynthesis is increased. But high light intensity, in presence of high light intensity, the chlorophyll molecules are degraded or destroyed. This process is known as photooxidation. The photooxidation of chlorophylls, the photooxidation of chlorophyll molecules under very high light intensity. The photooxidation of chlorophyll molecules under very high light intensity is known as solarization. So it is one of the most important ones. Solarization. So clear. So solarization. So light intensity. So first external factor is light intensity. The light intensity, the light intensity at which rate of photosynthesis is equal to rate of photosynthesis is known as compensation point. If light intensity is increased above compensation point, the rate of photosynthesis is increased. But high light intensity, but in presence of high light intensity, the chlorophyll molecules are degraded. This process is known as photooxidation. The photooxidation of chlorophyll molecules under very high light intensity is called solarization. Is called solarization. Usually, the rate of photosynthesis usually usually the rate of photosynthesis. So next one, the rate of photosynthesis 10 times. So 10 times more than respiration. Usually the rate of photosynthesis more than, so more than 10 times, more than 10 times more than respiration during daytime. During daytime, the rate of respiration 10 times more than the rate of photosynthesis more than 10 times more than respiration. But in the evening, but in the evening, at the point, at the point, the rate of photosynthesis is equal to respiration, is known as light light compensation point. Compensation point. Compensation point. So clear. Usually the rate of photosynthesis 10 times more than. Usually the rate of photosynthesis 10 times more than respiration during daytime. But in the evening, at the one point, the rate of photosynthesis is equal to rate of respiration is called light compensation point. It is not good for plant. It is not good for plants. It is not good for plant. So this is the first factor. The factors affecting this is the first factor, first external factor. So clear. This is the first external factor in the photosynthesis. So clear. So once again, so first external factor is light intensity. So light intensity at which rate of photosynthesis equal to the rate of respiration is called compensation point. If the light intensity, intensity is increased above compensation point, the rate of photosynthesis is also increased. But high light intensity chlorophyll molecules are degraded. This process is known as photooxidation. The photooxidation of chlorophyll molecules under very high light intensity is called solarization. Usually, the rate of photosynthesis 10 times more than respiration during the daytime. But in the evening, at the one point, the rate of photosynthesis is equal to rate of respiration is called light compensation point. So it is not good for plants. So clear. This is the first external factor of the photosynthesis. And next one is the next factor. So next one is the light quality. So light quality. So next factor is light quality. The rate of photosynthesis is higher in red. The rate of photosynthesis. The rate of photosynthesis is high. Is high. Is high in red light. Red light followed by blue light. Followed by blue light. The rate of photosynthesis is very high in red light, then after blue light. So it is minimum. So it is minimum. It is minimum in green light. So it is minimum in green light. And it is maximum. The rate of photosynthesis is maximum in white light. In white light. So white light, it is also known as polychromatic light. So polychromatic light. Polychromatic light. So clear. The light quality. The second factor is the light quality. The rate of photosynthesis is high. The rate of photosynthesis is high in red light and followed by blue. The rate of photosynthesis is minimum in green light because the plants do not absorb the green light, it reflects. So it is minimum in green and maximum in white light. So it is maximum in white light. So clear. Generally, the plants which can grow, generally, the plants which can grow, the plants which can grow, the plants which can grow shade areas, the plants which can grow shade areas are called cyophytes, cyophytes. Generally, the plants which can grow shades are known as cyophytes. Cyophytes have very sensitive. The cyophytes have very sensitive and efficient photosystems to receive diffuse light. The cyophytes have very sensitive and efficient efficient photosystems to receive diffuse light. The plants which can grow shades known as cyophytes. The cyophytes have very sensitive and efficient photosystems to receive diffuse light, as well as the plants can grow in direct sunlight. Direct sunlight. The plants which can grow in direct sunlight are known as heliophytes. The plants can grow on shades are known as cyophytes. The plants can grow direct sunlight known as heliophytes. The plants are of two types based on light quality. They are cyophytes and heliophytes. Cyophytes can grow shades. Heliophytes can grow direct sunlight. So clear. And final one, the rate of photosynthesis. The rate of photosynthesis is more in intermittent light. So it is more in intermittent light. So intermittent means discontinued or light in flashes. The rate of photosynthesis more in intermittent light. or discontinued light or light in flashes than continuous light. Continuous light. So clear. The rate of photosynthesis more in intermittent light or discontinued light or light in flashes than continuous light. So continuous light lo kante సో ఒక ప్లాంట్ పైన కంటిన్యూగా లైట్ పంపించిన దానికంటే సో డిస్కంటిన్యూగా లేదా ఫ్లాషెస్ గా లైట్ ను పంపించినప్పుడు ఫోటోసింథటిక్ రేట్ అనేది పెరగడం జరుగుతుంది. ఇక్కడ ద రేట్ ఆఫ్ ఫోటోసింథసిస్ మోర్ ఇన్ ఇంటర్మీడియట్ లైట్ ఆర్ డిస్కంటిన్యూ లైట్ ఆర్ లైట్ ఇన్ ఫ్లాషెస్ దాన్ కంటిన్యూస్ లైట్ బికాస్ ఆఫ్ ద అసిమిలేటరీ పవర్ అక్యుములేటెడ్. Because of ఎందుకంటే because the assimilatory power accumulated it is not used in dark reaction. So why the rate of photosynthesis more in intermittent light than continuous light? Because of the assimilatory power accumulated. It is not utilized in dark reaction. So the rate of photosynthesis is decreased. So clear. This is the second one. Second one is the light quality. So light quality. The light quality, the rate of photosynthesis. The rate of photosynthesis higher. The rate of photosynthesis higher in red light than after blue light. It is maximum. It is maximum in green light and minimum in white light. The plant which can grow shades known as cyophytes, they have very sensitive and effective photosystems to receive diffuse light. The plant which can grow in direct sunlight area are called heliophytes. The rate of photosynthesis is more in intermittent light or light of flashes than in continuous light because assimilatory power accumulated. It is not used in dark reaction. So clear. Our next factor of the photosynthesis. So next factor of the photosynthesis is carbon dioxide. So carbon dioxide, it is one of the most limiting factor. So carbon dioxide is one of the most common. It is one of the most common limiting factor. Is one of the most common limiting factor. The most common limiting factor is carbon dioxide, then after light. So limiting factor, first one is the carbon dioxide. So the most common limiting factor is the carbon dioxide. So carbon dioxide is one of the most common limiting factor. So limiting factor, then after is light. Generally the normal concentration. So normal concentration of CO2. The normal concentration of CO2 in atmosphere is about 0.03% or 330 ppm. So 330 ppm. The normal concentration of carbon dioxide in atmosphere is about 0.03% or 330 ppm. The concentration of carbon dioxide is increased. The rate of photosynthesis is also increased. The concentration of carbon dioxide is increased. The rate of photosynthesis also increased. The carbon dioxide concentration increased. The carbon dioxide concentration increased from 0.03 percentage to 1 percentage. The carbon dioxide concentration is increased from 0.03 percentage to 1 percentage. The rate of photosynthesis, so the rate of photosynthesis is also increased. The carbon dioxide concentration increased from 0.03 to 1%. The rate of photosynthesis is also increased. The carbon dioxide concentration increased more than 1 percentage. The rate of photosynthesis is decreased due to the stomata close, because of stomata closing. So clear. The one of the most common limiting factor of photosynthesis is carbon dioxide. Thereafter light. The normal concentration of carbon dioxide is 0.03 percentage or 330 ppm. The concentration of carbon dioxide increased. The rate of photosynthesis also increased. The carbon dioxide concentration increased from 0.03 percentage to 1 percentage. The rate of photosynthesis is also increased. The carbon dioxide concentration increased more than 1 percentage. The photosynthetic rate is decreased and stopped because of the stomata is closed. This is the second factor. Is called second one, carbon dioxide. And next to photosynthetic factor. The next photosynthetic factor is carbon dioxide compensation point. So compensation point. So already we know. So compensation point. So compensation point means the rate of photosynthesis. The rate of photosynthesis is equal to rate of respiration. Respiration is known as compensation point. So compensation point. Generally, it is 25 to, so the carbon dioxide compensation point, 25 to 100 ppm, or above 50, above 50, in C3 plants. In C3 plant, the carbon dioxide compensation point is 25 to 100 ppm or above 50. In C4 plants, the carbon dioxide compensation point is 0 to 10 ppm, 10 ppm in C4 plants. In CAM plant, the carbon dioxide compensation point is 0 to 5 ppm. So this is the one of the most important one. So carbon dioxide compensation point. The rate of photosynthesis is equal to rate of respiration is known as compensation point. The carbon dioxide compensation point in C3 plants is 25 to 100 ppm or above 50 ppm in C3 plants. In C4 plants, it is about 0 to 10 ppm or below 10. In CAM plant, it is about 0 to 5 ppm. So clear. And next to photosynthetic factor is temperature. So next photosynthetic factor is temperature. The optimum temperature for photosynthesis is 20 to 35 degrees temperature or 20 to 30 degrees temperature. The optimum temperature for photosynthesis is about 20 to 35 degrees or 20 to 30 degrees. So above 40 degrees temperature, the rate of photosynthesis, above 40 degrees, the rate of photosynthesis is decreased due to the denature of enzymes. The temperature is increased. The temperature increased above 40%. The photosynthetic rate is decreased because of denature of enzymes. At the same time, the above temperature more than 40, it decreases the photosynthesis. At the same time, the rate of photosynthesis is inhibited. It is inhibited at 0 degrees temperature. The rate of photosynthesis decreased above 40 degrees and below 0 degrees at 0 degrees, except the rate of photosynthesis inhibited at 0 degrees, except lichens, some lichens. Generally, the rate of photosynthesis inhibited at 0 degrees, except lichens, coniferans, 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 coniferans. They show photosynthesis even minus 20 to minus 35 degrees temperature. Generally, the rate of photosynthesis is inhibited at 0 degrees. But in some plants like lichens, coniferans, they show photosynthesis at even to minus 20 to minus 35 degrees. So clear. At the same time, the algae Oscillatoria brevis. The algae Oscillatoria brevis show photosynthesis at higher temperature. It is about 70 degrees temperature. Generally, the temperature increased more than 40. The rate of photosynthesis is decreased. The photosynthesis inhibited 0 degrees, but it occurred lichens and coniferans at the temperature minus 20 to minus 35. In Oscillatoria brevis, the photosynthesis even occurred at high temperature at 70 degrees. In C3 plants, the suitable temperature for C3 plants is about 20 to 25 degrees temperature. The optimum temperature of C3 plants is 20 to 25 degrees. In C4 plants, it is about 30 to 45 degrees temperature. So the optimum temperature, the suitable temperature for C3 plants is 20 to 25 degrees. In C4 plants, it is about minus 30 to 45 degrees. So next, another important one. So an important factor of the temperature. With an increase in temperature, with an increase in temperature, from 0 degrees to 35 degrees. An increase in temperature from 0 degrees to 35 degrees temperature. The rate of photosynthesis increases twice for rise of 10 degrees. So clear. This is one of the most important. It is called Q10. It is called Q10. With an increase in temperature from 0 degrees to 35 degrees, the rate of photosynthesis increased to twice. For rise of 10 degrees, it is called Q10. Generally, the Q10 value is 2. So remember it, the Q10 value is 2. So it is one of the most. So temperature no 0 నుంచి 35 degrees temperature ku 0 నుంచి 35 degrees కు 10 times. సో 0 నుంచి 10 ఒకటేసారి 10 నుంచి 20 20 నుంచి 30 అంటే ఫర్ రైజ్ ఆఫ్ 10 degrees. సో 0 నుంచి 35 కు 10 degrees పెంచుకుంటా పోతే సో 10 degrees పెంచుకుంటా పోతే ఫోటోసింథసిస్ రేట్ అనేది పెరుగుతుంది. దీన్నే Q10 అంటారు. ద Q10 వాల్యూ ఇస్ 2. సో దిస్ ఇస్ వన్ ఆఫ్ ద మోస్ట్ ఇంపార్టెంట్ వన్. సో వన్ ఆఫ్ ద మోస్ట్ ఇంపార్టెంట్ వన్ ఇస్ ద Q10 వాల్యూ. So Q10 value is the most important one. So clear. This is the information about the another factor is temperature. And next factor, oxygen. So next factor is oxygen. So next one is the oxygen. The concentration of oxygen. The concentration of oxygen increased. The oxygen concentration is increased. The rate of photosynthesis is decreased. The oxygen concentration is increased. The rate of photosynthesis is decreased. So it is called Warburg effect. So one of the most important one, Warburg effect. So it was first discovered by Warburg, hence it is called Warburg effect. So what is the Warburg effect? The rate of oxygen, the oxygen concentration is increased. The rate of photosynthesis is decreased. It is called Warburg effect because of in C3 plants, the concentration of oxygen increased in C3 plants. Photorespiration is occurred. So photorespiration is occurred because of the Rubisco, the enzyme Rubisco act as RUBP oxygenase at high oxygen concentration. The enzyme Rubisco act as RUBP oxygenase and perform the photosynthesis. As a result, the photosynthetic rate is decreased. The rate of photosynthesis is the concentration of oxygen is increased. The photosynthetic rate is decreased. So it is called Warburg effect because of the oxygen concentration increased in C3 plants. The enzyme Rubisco act as RUBP oxygenase and perform the photorespiration. As a result, photosynthetic rate is decreased. So clear. This is the another factor is called oxygen. And next one is the water. So another photosynthetic factor is water. So water deficiency. So water deficiency. So water deficiency close the stomata. So water deficiency close the stomata. So water deficiency close the stomata. As a result, the rate of photosynthesis is also decreased. The rate of photosynthesis also decreased. So clear, the water deficiency causes close of stomata. As a result, the rate of photosynthesis is also decreased. Next one, what is the importance of water? In photosynthesis, the water donate electrons and protons to NADP plus. So the water is the electron donor for photosynthesis. So water is the electron donor. So water is the electron donor in photosynthesis. Next one, photolysis of water. The photolysis of water takes place. The photolysis of water. Photolysis. So photolysis of water takes place at plus 0.8 volts. This is the one of the most important one. The photolysis of water takes place at plus 0.8 volts. So 0.8 volts. So clear. The rate of photosynthesis, is decreased when the deficiency of water because of close of stomata. So water play an important role in photosynthesis. The water donate electrons and protons to the NADP plus. The photolysis of water occurs at 0.8 volts. The plant utilizes. So next important one, the plants utilize less than the plant utilize less than 1% of water 1% of water absorbed by them in photosynthesis absorbed by them absorbed by them in photosynthesis. So ప్లాంట్స్ అబ్జర్వ్ చేసినటువంటి వాటర్ లో కేవలం 1% వాటర్ మాత్రమే ఫోటోసింథసిస్ లోపల యూజ్ చేయడం జరుగుతుంది. ఇది ఇంకొక ఫ్యాక్టర్. అండ్ ద ఫైనల్ ఫోటోసింథటిక్ ఫ్యాక్టర్ ఇస్ ద మినరల్స్. The final external factor of photosynthesis is the minerals. Generally, magnesium, iron, copper, manganese, chlorides, and calcium. These ions play an important role in photosynthesis. So minerals. In photosynthesis, the minerals like magnesium, iron, copper, manganese, chlorine, calcium, they play an important role in photosynthesis because the magnesium ion is needed for chlorophyll synthesis. It is needed chlorophyll synthesis. The magnesium Mg+2 is required for the synthesis of chlorophyll. And next one, iron. So iron, it is one of the most important component in pyridoxine. It is important component in pyridoxine and cytochromes. So iron is the important component in the pyridoxine and cytochrome. So copper is also important component in plastocyanin. The electron carrier plastocyanin. It consists of copper. So plastocyanin is the electron carrier. It consists of copper. And next one, Mn+2, Cl- and Ca+2. These ions important components in oxygen evolving complex, OEC, oxygen evolving complex. These are the main components of oxygen evolving complex. They play important role in photolysis of water. So photolysis of water takes place in presence of ions, manganese, chloride, and calcium. These are the factors of photosynthesis. The factors are affecting photosynthesis. All these are external factors. And second one is the internal factor. So internal factors. So already I mentioned the internal factors are chlorophyll content, protoplasmic factors, accumulation of products, and anatomy of leaf, etc. These are the internal factors affecting the photosynthesis. And the final one. So final one, major experiments. So major experiments in photosynthesis. Major experiments in photosynthesis. The necessity, important one, all these are important experiment. The necessity of carbon dioxide. The necessity of carbon dioxide for photosynthesis. For photosynthesis can be demonstrated by the necessity of carbon dioxide for photosynthesis can be demonstrated by moles of leaf experiment the moles of leaf experiment can be demonstrate the necessity of carbon dioxide for the photosynthesis. The necessity of carbon dioxide for the photosynthesis can be demonstrated by moles of leaf experiment. So clear. And next one, the necessity of chlorophyll. The necessity of chlorophyll. The necessity of chlorophyll for photosynthesis can be demonstrated by light screen experiment. So clear. The necessity of carbon dioxide for photosynthesis can be demonstrated by moles half leaf experiment. And next one, the necessity of chlorophyll for photosynthesis can be demonstrated by light screen experiment. So clear. And next one, the liberation of oxygen. Liberation of oxygen during the photosynthesis. During the photosynthesis can be demonstrated by hydrilla funnel experiment. It is demonstrated by hydrilla funnel experiment. The liberation of oxygen during the photosynthesis can be demonstrated by hydrilla funnel experiment. So clear. And next one, the presence of starch. The presence of starch can be tested by iodine test. So iodine test is used to demonstrate the presence of starch in the plant. The presence of starch can be tested by iodine test. The liberation of oxygen during the photosynthesis can be demonstrated by hydrilla funnel experiment. The necessity of chlorophyll for photosynthesis can be demonstrated by light screen experiment. The necessity of carbon dioxide for photosynthesis can be demonstrated by moles of leaf experiment. So clear. And next one, the rate of photosynthesis can be can be known by counting air bubbles. So counting air bubbles of oxygen. Counting air bubbles of oxygen through Wilmot's bubbler. So clear, the rate of photosynthesis can be known by counting air bubbles of oxygen through Wilmot's bubbler. Next one, the light is essential for photosynthesis. The light, the light is essential. The light is essential for photosynthesis. Can be proved by, can be proved by, can be proved by Gauguin's, Gauguin's light, proved by Gauguin's light screen experiment. Light screen experiment. So clear. So these are the major experiments in photosynthesis. So first one is the necessity of carbon dioxide for photosynthesis can be demonstrated by Mole's half-leaf experiment. And next one, the necessity of chlorophyll for photosynthesis can be demonstrated by light screen experiment. The liberation of oxygen during photosynthesis can be demonstrated by hydrilla funnel experiment. The presence of starch can be tested by iodine test. The rate of photosynthesis can be known by counting air bubbles. Counting air bubbles, counting air bubbles of oxygen through Wilmot's bubbler. The light is essential. The light is essential for photosynthesis can be proved by Gauguin's light screen experiment. Light screen experiment. These are the main important experiments. Main important experiments in photosynthesis. So clear. So this is the major topics. So these are the major topics in the photosynthesis.